And next is Rebecca Mobley. And Rebecca, we ran into a year and a half ago. Actually, I guess you've been talking to Southampton and therefore to us for almost 2 years, I think, right? And Rebecca has the distinction of being our first fellow, our first research fellow. So she and the University of Southampton are the recipients of the first PURA grant for research and studies. Now, I want to be careful about that. That grant did not fund her 100%, or her study, which was between 12 and 18 months, and we're at 12 months now, or 13 months now. We actually teamed up with the university as well as another private donor, and three parties put money into her research project and her ability to put this all together. And we're very honored to have her. She also has a doctorate in medicine, so for pediatrics. And so that was a really great combination because it's, so many of our new incoming patients are adolescents. It's, as we grow older, we are finding more and more adult patients, but by and large, we're a younger community that is being identified right now. So she certainly had a certain insight and understanding to some of that. So, Dr. Rebecca Mobley, thank you.
Thank you. So I think I've got the penultimate session of today, so I will try and keep this as interesting and as short and sweet as possible. But this has become a bit of a baby of mine over the last year. So, and I think everyone at Southampton and the foundation are quite proud of it. So hopefully I'll be able to sell it to you all today, because ultimately we are really relying on you to make this successful. So what I'm going to talk about is the PURA syndrome longitudinal natural history study, which has become synonymous really with the registry. I know many of you call it the registry. And for ease, I think I'll probably call it the registry for the rest of this presentation. But what I want to talk about today is what we know so far about PURA syndrome, and I know Helen spoke a lot about that, so I won't go into that in huge amounts of detail, but what I do want to talk about is how that's given us momentum to conduct this study, really. And then I'll talk a bit about the study, where we're at, and how you will get involved, which I really hope that you really want to and do. Just to start the PURA story, so on the left there's a swan there, and I don't know how many of you kind of know about the swan. I'm sure you do, but before 2014 many of you had children where you knew that they had a genetic diagnosis, or you knew they had a syndrome but you just didn't have a diagnosis for that. And we call that in the UK, and I think probably in the US you do as well, a syndrome without a name, and so that's why the swan's there. But then in 2014 a lot changed, and two papers came out in really close succession. One actually from the University of Southampton, David Hunt and Diana Borelli, who we've heard a bit about today. And they described the first patients with PURA variants and an associated disease or syndrome, which we now know as PURA syndrome. And now, before 2014, there was a lot of information about PURA, as you've heard today, about the mouse and the protein, and obviously the 5q31.3 microdeletion syndrome. But 2014 was the first time that patients with PURA variants, that coding change in the PURA gene and the PURA syndrome, the neurodevelopmental problems, the intellectual disability, the poor tone was described. And since then, really, the literature's grown. So 2015, we had another paper, Tanaka, and this paper didn't really expand the phenotype so much. He didn't add any more characteristics. But what they did do is they added different variants. So what's really important is, just because you have a change in your PURA gene, it doesn't mean that you have PURA syndrome. It's got to be a specific place. And I'm sure there's many children who have many different variants in this room. And what we want to know is which ones do cause disease and which ones don't. And so these studies have added more and more variants as we've gone along the road. And then 2017, a really busy year. But this big paper, this 32 paper, Margot's paper here, added some more patients. And really importantly, what this allowed us to do is identify new phenotypes, new patient characteristics. So this is when, you know, the excessive sleepiness was described, the short stature, the hiccups in utero. And I think for some of you, that probably was a bit of a eureka moment. Oh, actually, that fits, you know, that fits in that jigsaw puzzle. And then the first case reports started coming out, those really rare things that might be associated with PURA syndrome, such as premature thalarchy, so breast development. And that helps direct research, really. And in 2018, some more case reports. So where have you gone, basically? I mean, it's really impressive. 2014, you had 15 patients in those orange countries. 2019, you have over 300 patients in those blue countries. You've got a person with PURA syndrome in almost every single continent in the world. And I think that, you know, I think what you've done for PURA syndrome is quite amazing, and having all these meetings and a global network and things is really quite inspirational. But so you've gone quite fast. So why am I telling you all of this? I'm going to tell you about the study, but what I want to show you is in 5 years how much momentum you've got from learning. And this hasn't just been a small learning curve, it's been a big learning curve, and there's, I think, as you've heard today, we've got a lot more learning to do. But I think I just wanted to raise some of the things that I think hopefully you've learned, or we've learned, and then why this has given us momentum to collect more information, which is the registry. So I really like this quote from Marie Curie, which I just wanted to share with you. Obviously, she's a Nobel Prize winner, and she won it for her work on polonium, but she said, "Nothing in life is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less." And that's what I think we want to do. We want to understand more. So how has this learning helped us so far? Ooh. So first of all, hopefully it's helped you, it's helped you, and you go from, I know that it's very difficult when I see children without a diagnosis, you know, not having access to certain facilities and care, and that sometimes having a diagnosis, how those things can help, and then learning more from it. And this was a really good paper from Rare Disease UK, which summarised some of the key findings. And one of it was saying that the families really want more information, and I think that's something that we really want to do, and I think it's something that we started doing, but something to be expanded upon. The information so far has allowed Margot to write, and her collaborators to write, the first gene reviews article, which basically also had some surveillance, evaluation and management guidelines, and that's brilliant for clinicians, especially in rare disease. But hopefully it's useful for you as well, because I think sometimes you need to advocate for your children, and I hope that you don't feel like you have to, but sometimes it is necessary, and hopefully these things can help you do that, you know, set a standard of care. It's helped us learn about all those quirks of PURA syndrome, you know, and as I said, we collected more and more individuals and patients, we learned about their Mr. Sleepiness and their hand movements and their short stature. And not only does that, as I said, help hopefully piece towards a big jigsaw puzzle of PURA syndrome, it also really helped with diagnoses. So at the core of PURA syndrome, with the neurodevelopmental problems, the seizures and the low tone, sometimes that doesn't set it away from other syndromes, and sometimes having some of these other quirks help you identify these patients. And I know that most of you in the room will have been diagnosed what we call a gene agnostic approach, where you make a diagnosis because you look for every single gene, whereas actually in Margot's paper, there's patient 11. Sorry, that hasn't come out very well at all, has it? And this patient was actually diagnosed by single gene testing. So someone actually saw this chart and thought they've got PURA syndrome and tested for that. And you know, we didn't even know about this syndrome 5 years ago. So I think it shows how things have changed. And what I was talking about is collecting those variants. There's a big push towards collecting these variants and helping with clinical diagnoses, and things like Decipher allows clinicians to share the variants and make good diagnoses, really. So I'll talk about the study now. So what is it? It's a longitudinal natural history study. Longitudinal, we're collecting data over time. Natural history is observational. We're watching what happens. Why have we not just called it a registry? So I guess a natural history study has a really firm goal. You really want to watch what happens over time. You want to see variables, which is what you were talking about earlier. You want to see how things change and interact. A registry is generally just a collection, storage, dissemination of information. But the brilliant things with registries is they have huge functionality. So what we have applied for and the ethics that we've got for this is to conduct it as a study but have the functionality as a registry as well. We're going to include any person with PURA syndrome, the deletion of the gene or the duplication of the gene. It's an international study. We are collecting information by means of online questionnaires, and I'll show you some of those later. And we're collecting it, importantly, from families and clinicians, because we really think that you can tell us different things. It's a specifically spec'd PURA study platform, and we really want to disseminate the study data for the purpose of research. We're not going to just hold on to this, we really want to share it. But I'll caveat that with, we want to share it safely, so we're not going to be selling your data or anything, but what we do want is we want people to be interested in it. We want this to be a really good data set so that people come and are interested in researching PURA syndrome. And it's a University of Southampton and a PURA Syndrome Foundation collaboration. So the University of Southampton is sponsoring it, and Diana Borelli, who Andrew spoke about in his talk, is the chief investigator for it, who's a professor at the University of Southampton. So why do we want to do this? I mean, I think I spoke about what we've learned about PURA syndrome so far and what else we want to learn, but we really want to understand the full spectrum of PURA syndrome. We want to know the full phenotype to help with diagnoses, to help with management, to help with surveillance. We want to understand the progression. We really want to understand the prognosis, which I really don't think we know huge amounts about at the moment. We want to identify possible phenotype-genotype correlations, which is kind of what we've been talking about all day. We want to understand why there's variability between the patients or the children, and whether this is because of where the change is, where the variation is, where the mutation is. And that's really important, especially for our scientists, but also for kind of, again, surveillance management guidelines. We want to formalise the collection of information internationally. We really don't want to fragment you. We want to keep you all in one place, and we want to get a really good data set. We want, as I said, we want to facilitate external research. This is a real patient-centred study, so we want to identify major quality of life issues for you, and we want to collect information that you want collected and you see as priority information. And we want to use this also as a platform to update you about other research opportunities. So you are in no way obliged to partake in any other research if you're part of the study, and actually if you don't like the study you can leave at any point. But what we hope is that we can also circulate information to you about other research opportunities. So just a quick overview about why some success stories. I mean, there's loads about natural history studies and registries, but so the top one was actually it was a paper about how they'd used registries and putting all your information together to recruit people for clinical trials, and it had shown that it was a really good, efficient system, and it was things that a lot of the drug companies liked, and a lot of the big other trials liked as well. The bottom one was looking at the natural history of mucopolysaccharidosis, which is a neurodevelopmental condition. It's a regressive condition. It's a storage metabolic problem. And it identified by collecting data, it could use imaging as prognostic markers. The top one is, it was about a behavioural profile in Rett syndrome, which I think forms a differential diagnosis for some of your children because of the hand movements and the neurodevelopmental phenotype. And they identified different behavioural profiles, and they said how they could, using these different profiles, they could use them to then make a distinction between successful and non-successful treatment trials. And the bottom one, which is really quite fascinating, was about this medication called asophosphatase, which was used in hypophosphatasia. And what they did is they used the natural history study, a historic natural history study, and then a history study of a recent patient cohort who'd had treatment and compared outcomes. And that allowed them to get this drug orphan registered, which essentially is a way of promoting drugs to be kind of pushed through that are going to be used for rare disease. So there's lots of ways that they can be beneficial. So who's involved? Well, obviously you're all involved, hopefully. And so we've got our children and the families and your clinicians who hopefully we hope to collect data from. And then at the University of Southampton, we've got Diana Borelli, who's the chief investigator and the data custodian, and really has been a driving force in this. She's had a lot of enthusiasm for it. And Richard Munday, who works at the clinical informatics unit and research unit in Southampton with Nicole and Kevin, and they basically made the software specific to PURA syndrome. So that's who's at the University of Southampton. Obviously, the PURA Syndrome Foundation have been really involved. They have checked, rechecked again all the information, all the collaborative agreements. I mean, there's something like 30 study documents that made up the application for this study. So, and talking about Mel, she was also really instrumental in writing the first lots of drafts of the questionnaires and also the ethical approval. The Global Research Network and their collaborators have also been involved in helping writing some of the study material. Alea is the software that we're using to hold all the data. It's all owned by someone called Forms Vision, which is a Dutch company. The University of Hospital Southampton are going to help us with getting people onto the study system and some of the administration. And then there's the PURA Syndrome Advisory Steering Committee, who are going to basically oversee the data application requests and promote that for external research. So where is this all happening? So I said it was an international study. I have to caveat that with some of these countries in red we've had to exclude because of UK and US sanctions. But Dominic was saying that actually I think we've only got 2 or 3 patients that are actually in those countries. So we really do want to try and get as many patients, families, clinicians involved in this. It's going to be available in 8 languages, although this is likely to be phased in because, as you can imagine, 28 documents that need translating in 8 languages, it's going to be quite hard work and quite time consuming. Then what we hope is that you'll all add your information online. It's all encrypted in transit. It then goes to a server. Then the software used is this Alea software. And then it goes onto a server in the Netherlands, which is owned by someone called Interconnect. And these companies are all have to follow data legislation. They all are have got all the high information security, redundancy packages if something overheats. I mean, they've got everything basically. So, and just a quick plug for rare disease, because I think we've got to remember that PURA syndrome obviously is a rare disease, and how you design your study is obviously needs to take that into account. And I really like this quote. It's from NCAT, which is a US National Center of Advancing Translational Science, by RADAR, which is their program to basically help rare disease registries be developed. And what they said is there's power in numbers. Each person's story matters. Registries combine a patient's community's stories into quality data that can lead to new discoveries and research advances. And I really think that's quite important. And Margot spoke about this last year. She said that she wouldn't have asked the questions about things like hiccups or the sleepiness until you'd all given her the clues. And we're really hoping that you give us some more clues so we can collect some more information and see if it's just your child that's got an individual quirky trait, or actually all of your children share that. So, quick plug for rare disease research. So there's no real guidelines on how best to go about starting a natural history study, but there's lots of advice, and basically I've tried to collate all of those papers that we spent hours reading into a slide, and these were the things that I thought were quite important and what we've tried to put into our study. So we want to ensure there's no duplication, and we know that there's not. We want to encompass the widest geographical area, which we are hoping to do. We want to avoid fragmentation of the patient population, which I think is probably what I've just said. We want to have a minimum set of common data elements, and we've really tried to do this, although it's still these common data elements are still very preliminary in rare disease research. So a lot of them apply to registration, so when you put your information, but not actually information about the rare disease. So we've put in as many of these as possible. We want to make it usable for you, and I think if I can use a system, then anyone can use a system, because I'm not great with computers, so hopefully it's okay for you. We want to, it has to be data, it's got to be ethical, it's got to follow legislation, it's got to be, and it's gone through all of that, and it's been a year in the making, and that's really been why. We want it to be patient-centred, and we need to optimise safe data sharing, which is what we've tried to do. So, how do you get involved? So just where is the study at, really, I guess, is we've got English ethical approval, we're waiting for Scottish ethical approval, we've got a meeting at the end of the month, and then hopefully we'll be able to get it out to you, really, and that's for international. For the clinicians, it's slightly different, and we'll be able to talk to them about it. So how do you get involved? So once we get the green light, we're going to put it all over the PURA Syndrome Foundation website. We then ask that you email us with some demographic information, which will say what that needs to be. We'll then check that you're eligible, i.e. that you don't have an account, that you're not in any of those red countries. We will then give you some login details, and then, so this is when you log into the system. And then you can choose your language. And then the consent form should become available. It's a bit longer than that, and then once you've signed the consent, you can see the study material. And then at the moment, there are 7 questionnaires, which I'll show you in a second. And then we'll ask you to annually update that information, because ultimately we want to see how this changes over time. We also want the clinicians to get involved, and there's 2 ways of getting them involved. And we think that probably, because you're such an active foundation, that it's probably going to be more by this means. So we ask that when you sign the consent form, as part of the consent form, it's for us to be able to contact the named clinician, and then you provide us with the clinician details, and that's all on that study form. We will then contact them, and if they are happy to proceed, we'll then give them login details. And then they don't have a consent form, but they have a study terms form, which just basically means that they follow some basic data holding and entry level, and also that they've got the appropriate institutional and legislation to enter information onto the system. And then the other way is if you haven't got access and the clinician recommends you to enter information about your child. So, and if this is the case, obviously you won't have had access to the online information. So they will have hard copies of the consent form, which we have got, and we've got through ethics as well. So the study flowchart is quite complicated in the protocol, and I've actually got the protocol with me. It's quite long, but if any of you want to read it, then I can share it with you. So these are the 7 documents, the 7 questionnaires, so genetic and family, pregnancy and birth. So that's going back to what you're talking about. Was there anything that you all share that has led to this? I think it's probably not, but there's no harm collecting the information. Development, neurology, and then if your child has seizures and this seizure one will become available, growth and endocrine, which is something that I've got quite a lot of interest in, and especially with puberty and bone health. And if you don't want to answer questions because you just don't feel that they're appropriate, then you just don't have to answer them. Every single question has a not sure or do not want to answer. So don't feel like you have to. But what we do ask is that you enter non-speculative information. So if you don't know, you don't know. That's fine. But try not to make it up. And the clinicians one, same questionnaires, but different questions. So how are we going to share the information? And I suspect that might be a worry for some of you. So it is a formalised system. If someone wants to use the information, they've got to apply for it. And then this is discussed at this steering committee that I was telling you about earlier. They then give an opinion. If the opinion is favourable, then data is provided. And then hopefully those data findings will be published. All of the researchers who've got data from this data set you'll be able to see, there'll be a list of them available on the PURA syndrome website, foundation website. And I've simplified that slightly because actually it's quite a few more steps, but that's the basic principles. But again, I mean, you can see the flow sheet next to it, so that's all in the protocol. The information that we'll be giving to researchers is de-identified, so we won't be giving them your child's name, we won't be giving them your child's date of birth, we won't be giving them your address or your contact details, but I can't say that it's going to be fully anonymous because there might be only one child with one variant, and so, you know, and they might put that variant in a paper, so you might be able to identify your child from a paper, but I mean, other people won't, but you know, your clinician or you might be able to. But we will try and keep it as anonymous as possible. So just a quick plug about data, because I think that's what I spent about 6 months doing. But it's GDPR compliant, which is the General Data Protection Regulations in the EU. It's a massive piece of EU legislation, and it's also been through a data protection impact assessment at the University of Southampton. There's a patient information leaflet which clearly describes how we want to use the data, how it's going to be handled, and your data rights, because you do have data rights. As I said, it's going to be pseudonymized. The study platform access is strictly monitored and it's also audited. And for us to be able to give researchers your data, they've got to sign an agreement and which stipulates certain data usage, publications, and they've also got to acknowledge us and you, because I think you've all entered your data and spent hours probably doing that, and we really want you to be acknowledged for that. So you can see that it's been a long time. So I mean, I've only been here for a year, and this has been going on for a couple of years. So I think I came in maybe about here. This has all been about kind of getting ethics, getting all our data right, setting up the study. But we're so nearly there, and I really hope that we'll be able to open it in the next couple of months, really. Just to show you where I am and where some of those stops have been, some of those hurdles. So just to end, another quote from NCAT, quite like them. This is the director who's a geneticist, but he says, "I hear every day how alone patients with rare diseases and their families can feel, and how alone researchers studying rare diseases feel. So let's all be alone together and think about rare disease as a gigantic and ultimately beautiful jigsaw puzzle," which I think it is. So just some acknowledgements to you, the foundation and the family. Thanks for having me. To my colleagues at the University of Southampton, Diana, Justin Davies, who's a paediatric endocrinologist, David Hunt and Andrew, the Clinical Informatics Research Unit, who they've put a lot of time into making this PURA specific, this study platform, and they've been doing this all on the side to their normal work commitments. So Kevin Wheeler, Nicole and Richard. The foundation committee, and I was saying about Mel and Cecile, and also now the active committee who've really helped and really provided some feedback, but they've also told us what you want to know. So all of those questionnaire topics have been based on feedback that you've given them and that we've then written. So the global network and also all the collaborators who've also helped with the writing of study material, such as Margot and Rick Leventer. So I think I'm done. Any questions?